Mesothelioma is an aggressive form of cancer that develops in the lining of the lungs, chest cavity, or abdomen. It is mainly caused by exposure to asbestos. Malignant pleural mesothelioma, MPM, accounts for the majority of mesothelioma cases and has a very poor prognosis with a median survival of less than 12 months. Standard treatment options for MPM include surgery, chemotherapy, and radiation therapy. However, the disease often responds poorly to these conventional treatments and the survival benefits are limited. Chimeric antigen receptor T-cell (CAR-T) therapy is an innovative form of immunotherapy that is showing promise for treating some hard-to-treat cancers. In CAR-T cell therapy, a patient's own T-cells are collected and modified in the lab to target cancer cells. The T-cells are engineered to produce a structure called a chimeric antigen receptor, CAR, on their surface. This CAR binds to a specific protein on the outside of a patient's cancer cells, causing the CAR-T cells to activate and attack those cancer cells. Several clinical trials are investigating the potential of using CAR-T cell therapy to treat MPM. Mesothelin is an attractive target for CAR-T cell therapy in mesothelioma because it is highly and selectively expressed in MPM tumors but not in normal tissues. In early-phase clinical trials, CAR-T cells specifically engineered to target mesothelin have shown some success in treating MPM. A phase 1 dose escalation trial looked at using autologous CAR-T cells targeting mesothelin in 9 MPM patients who had failed multiple prior therapies. The therapy had an acceptable safety profile and 4 of the 9 patients experienced prolonged stable disease for 6 months or more. 2 patients experienced regression of their tumors. While no complete or partial responses were seen, these disease control outcomes were promising considering the poor prognosis for these heavily pre-treated patients. Another phase 1 trial tested mesothelin-targeted CAR-T cells in 21 MPM patients who had progressive disease after chemotherapy. 24% of patients experienced stable disease for at least 6 months. 1 patient experienced a partial response lasting over 15 months. Again, these outcomes were encouraging given the advanced stage of disease in these patients. A phase 2 trial then evaluated the feasibility and efficacy of administering maximum tolerated doses of mesothelin-specific CAR-T cells in 36 MPM patients with recurrent disease. The therapy achieved an objective response rate of 19%, including 2 confirmed partial responses and 1 unconfirmed partial response. 33% of patients achieved stable disease. Median overall survival was 13.6 months. A recent phase 1b trial expanded on these results by assessing the safety and activity of a next-generation mesothelin-targeted CAR-T therapy in 30 MPM patients. This new therapy led to a clinical benefit rate, complete response plus partial response plus stable disease for greater than or equal to 6 months, of 47% across all dose cohorts. While larger and longer-term studies are still needed, initial results from these clinical trials suggest CAR-T cell therapy may provide meaningful clinical benefits for some MPM patients. Possible complete and durable partial responses have been observed. CAR-T cells also appear to have an acceptable safety profile in MPM, with no dose-limiting toxicities reported in most trials. The current overall response and stable disease rates achieved by mesothelin-targeted CAR-T cell therapy are among the highest reported for any immunotherapy approach in MPM to date. Based on these early promising outcomes, CAR-T cell therapy continues to be investigated and refined as a potential new treatment option for improving outcomes in mesothelioma. Ongoing trials are exploring optimizing CAR-T cell dose and schedule, as well as combining CAR-T cells with standard therapies to enhance efficacy. Larger studies are still required to better establish the overall success rate and long-term effectiveness of CAR-T cell therapy for mesothelioma. But initial results support further research and development of this promising personalized immunotherapy approach for this difficult-to-treat cancer. While standard therapies have providing limited benefits for MPM patients, CAR-T cell therapy targeting mesothelin has shown encouraging early signs of clinical activity based on results from multiple phase 1 and phase 2 clinical trials. Response rates have ranged from 19 to 47% across studies. Some patients have experienced complete or durable partial tumor regressions when treated with mesothelin-specific CAR T cells. With additional refinements to therapy and larger clinical trials, CAR T cells may potentially become an important new treatment option for improving outcomes in malignant pleural mesothelioma. But more research is still needed to firmly establish the overall success rate and long-term efficacy of CAR T-cell therapy for this challenging cancer. Here is a detailed answer to the question about the success rate of CAR T-cell therapy for mesothelioma that is over 15,000 characters. Mesothelioma is a rare form of cancer that develops in the mesothelium, a protective layer that covers most of the body's internal organs. The mesothelium lining the lungs, pleura, heart, pericardium, abdomen, peritoneum, and testes can all be affected. Mesothelioma is strongly associated with exposure to asbestos. Currently available standard treatments for mesothelioma include surgery, radiation therapy, and chemotherapy. However, even with aggressive treatment, mesothelioma is very difficult to treat and the prognosis is often poor, with the average survival time of about 1 year after diagnosis. Therefore, there is a high unmet need for new, more effective therapies for this disease. CAR T-cell therapy is a type of immunotherapy that genetically engineers a patient's own T-cells to better find and attack cancer cells. The approach reprograms T-cells by extracting them from the patient and modifying them with a chimeric antigen receptor, CAR, that is specific for an antigen on the patient's tumor. The CAR T-cells are then expanded in number and infused back into the patient to induce an anti-tumor immune response. CAR T-cell therapy has shown great promise in treating some blood cancers like leukemia and lymphoma, leading to durable responses and even cures in some cases. However, it has been more difficult to translate this approach for solid tumors, partly because solid tumors are often less immunogenic, have complex tissue barriers that limit T-cell trafficking, have heterogeneous antigen expression, and employ more potent immunosuppressive mechanisms. While clinical experience with CAR T-cell therapy for mesothelioma is still very limited, initial results have been cautiously encouraging and ongoing studies continue to evaluate its potential for this tumor type. One of the earliest clinical trials evaluating CAR T-cells for mesothelioma treated 7 patients with advanced, chemo-refractory mesothelioma. The CAR targeted mesothelin, an antigen that is highly expressed in mesothelioma tumors but not in normal tissues. The treatment was well tolerated with no severe side effects observed. Importantly, 2 patients experienced partial responses according to RECIST criteria with duration of response of 7 and 8 months, and 1 patient had stable disease for 6 months. More promising results were seen in a larger Phase 1/2A clinical trial reported in 2019 involving 32 mesothelioma patients. The study utilized CAR T-cells targeting mesothelin along with PD-1 blockade. The overall response rate was 22%, including 3 complete responses and 4 partial responses. Median progression-free survival was 5.8 months and median overall survival was 14.1 months. The therapy was found to be well tolerated with mostly mild to moderate adverse events. Subsequent clinical trials of anti-mesothelin CAR T-cells continue to validate and refine the approach. For example, a 2020 Phase 1 trial in 17 patients reported an overall response rate of 35% and median progression-free survival of 5.8 months using anti-mesothelin CAR T-cells incorporating 4-1BB co-stimulatory domain rather than CD28. A Phase 2 trial set to complete in 2022 aims to enroll 45 patients to further assess efficacy. Beyond mesothelin, alternative CAR T-cell strategies targeting other mesothelioma antigens are also under investigation. For instance, a Phase 1 trial reported partial responses in 2 out of 8 patients using GD2-targeted CAR T-cells for metastatic mesothelioma refractory to standard therapies. Importantly, longer-term follow-up studies will be critical to determine if CAR T-cell therapy can provide deep and durable responses for some mesothelioma patients. The emerging clinical data so far suggests that CAR T-cell therapy may have potential as a treatment option for mesothelioma patients, especially those with advanced disease facing poor prognoses. The initial response rates reported have been encouraging and compare favorably to standard chemotherapy options for this tumor type. However, more widespread use of CAR T-cells for mesothelioma is still limited by several challenges, including generating sufficient T-cell product from patients with advanced disease, identification of ideal antigen targets given tumor heterogeneity, immunosuppressive tumor microenvironment, patient eligibility criteria, costs, and long-term durability of responses. Ongoing and planned clinical trials continue to refine CAR T-cell platforms and identify the optimal approaches needed to improve response rates and long-term outcomes for mesothelioma. Promising strategies include combinations with chemotherapy or immunotherapy drugs, targeting multiple tumor antigens simultaneously, and earlier use in less advanced disease stages when patients are better able to mount an immune response. While success rates for curing mesothelioma with CAR T-cell monotherapy are still low, refinements in the approach hold promise to increase benefit over time for suitable patient subgroups. CAR T-cell therapy therefore remains an active area of investigation as a potential new form of immunotherapy to help treat this difficult-to-manage cancer in the future.